Welcome to The Explainer. Today, we're looking at a totally fascinating ecological detective story. We're talking about saving the spice, specifically nature's microscopic bodyguards. Let's get right into it.
So, if you're anything like me and you love a dash of cinnamon in your morning coffee or, baked into holiday treats, you absolutely need to know what's going down in Vietnam right now. They're one of the world's top exporters of Cinnamomum cassia. That's the tree that gives us the cinnamon we all know and love. But starting around early 2017, this devastating outbreak just started tearing through their plantations. It's this aggressive disease called root rot dieback, and it's brutal. It basically rots the tree's roots, makes the bark peel right off, and eventually the whole tree just wilts and dies. The culprit behind all this? These highly virulent pathogens from the genus Phytophthora.
Okay, let's dive into this and see exactly what this invisible killer actually does. Researchers grade this root health on a scale of 1 to 5. Now, looking at a 1, that's your baseline, a completely healthy, robust root system. But as you move up that scale to a 3, a 4, and finally a 5, wow, you're looking at the visceral devastation these Phytophthora pathogens leave behind. By stage 5, literally 76 to 100% of the root system has been turned into this dark brown or black rotting husk. There's just no way the plant survives that. Now, you might be wondering, well, why not just blast the soil with chemical fungicides? I mean, they exist, right? But here's the catch. They come with some serious baggage. First off, they're incredibly expensive for farmers. Plus, they cause massive collateral damage to the surrounding environment. But the real kicker for us consumers, we use cinnamon bark directly as a spice and even as medicine. Spraying toxic chemicals leaves behind these unwanted residues that, let's be honest, nobody wants anywhere near their food. So to save the cinnamon industry without poisoning the actual product, researchers realized pretty quickly they needed a biological savior instead.
And that right there brings us to our microscopic heroes, endophytes. So what exactly is an endophyte? It's basically a microorganism, usually a fungus or a bacterium, that just lives its entire life inside the tissues of a healthy plant without making the plant sick at all. Think of them like nature's internal immune system, or, microscopic plant bodyguards. These little hidden tenants are amazing. They can produce compounds that actually promote plant growth, or even better, they can literally pump out natural bactericidal and fungicidal chemicals to fight off invading pathogens.
So how do we harness these tiny bodyguards? Well, a team of researchers in Vietnam put together this brilliant, super clear 3-step methodology. Step 1, trek into these diseased plantations, find the rare, completely healthy cinnamon trees that somehow survived, and isolate the natural endophytes living inside them. Step 2, take those isolates and pit them head-to-head against the deadly Phytophthora pathogens in a controlled lab setting. And finally, Step 3, take the ultimate winners of that lab battle and test them out in real, living cinnamon seedlings to see if they can actually protect plants in the dirt.
The whole investigation kicked off by rounding up the initial lineup of suspects. Researchers went out and collected healthy branches and leaves from 4 major cinnamon-producing provinces across Vietnam. And they were super meticulous about this. They completely surface sterilized all the plant tissues because they wanted to make absolutely sure they were only extracting the microbes living inside the plant, not just hanging out on the surface. In the end, they successfully isolated an impressive 134 microorganisms in total. That breaks down to 110 fungal isolates and 24 bacterial isolates. And the hope was that just one of these 134 suspects might hold the key to stopping the root rot.
Let's move to and see how this builds. Because with 134 unique endophytes ready to go, it was time for Trial 1, the in vitro screening. Or, as I like to call it, the Petri dish showdown. Here's the setup. The researchers took 3 of the absolute deadliest species of Phytophthora, the ones causing the worst root rot, and put them on agar plates. Then they dropped a single endophyte isolate on the exact opposite side of that same dish. Then they basically turned off the lights and waited 10 days to see exactly what would happen when these 2 organisms clashed.
And this brilliantly illustrates a literal microscopic turf war. Seriously, on these plates, you have the pathogen desperately trying to grow outward, and the endophyte is just actively fighting back. It creates this physical barrier to block the pathogen's spread. The researchers measured exactly how much the pathogen was able to grow in these dual cultures and compared it to control plates where the pathogen just grew totally unopposed. Basically, they weren't just looking for endophytes that could merely survive. They wanted the ones that aggressively push back and starve the Phytophthora out of its territory. Well, out of those initial 134 suspects fighting it out in the Petri dishes, the vast majority actually got eliminated. Only 17 elite endophytes proved they were tough enough to move forward.
To make the cut, these microbes really had to show up. Specifically, they had to demonstrate more than a 45% inhibition of the pathogen's growth in the lab. So these 17 finalists, which were this awesome mix of powerful penicillium fungi and some incredibly beneficial bacteria, survived to face the ultimate test.
Because, look, beating a pathogen on a flat, controlled agar plate in a lab, that's one thing. But doing it out in the complex, totally messy environment of living soil, that is an entirely different ballgame. Trial 2 escalated the stakes drastically with real-world greenhouse tests. The researchers took those top 17 microbes, and now they had to prove they could actively protect living, breathing cinnamon plants that had been deliberately infected with those deadly root rot pathogens.
Now, to figure out the most practical way for actual farmers to use this out in the field, the researchers ran 2 parallel tests to see the absolute best way to deliver these microscopic bodyguards. In the first track, they took naked cinnamon seeds and inoculated them with endophyte spores right as they were germinating. Then they followed up by spraying the tiny month-old plants. In the second track, they took a different approach. They waited until they had established 6-month-old seedlings from a nursery, and then applied the endophytes by spraying the leaves and watering the soil. Once the bodyguards were fully moved in and established, that's when they finally introduced the deadly Phytophthora to the roots.
Now, what's really interesting about this slide is just how clearly the numbers prove the sheer power of biological control. Look at these root damage ratings. Remember that 1 to 5 scale we talked about earlier? That massive black bar on the far right of each graph? That's the control group with absolutely no endophytes, and as you can guess, their roots were just utterly destroyed by the disease. But check out the bars for the inoculated plants. Just having those specific elite endophytes present drastically reduced the height of those dark bars across the board. The bodyguards were actually doing their job. They were actively keeping the root rot at bay in living, breathing soil. But wait, the researchers found something even more mind-blowing during these greenhouse trials. Not only did the absolute best candidates stop the rot dead in its tracks, but they actually triggered this amazing secondary benefit. I'll read this directly from the study. 3 endophytes expressing high levels of disease suppression and plant growth promotion. So actually think about what that means. The best of the best didn't just act as defensive shields. They acted like fertilizers. The cinnamon plants protected by these specific endophytes literally grew significantly taller and stronger than the control plants that had no pathogens near them at all. Crazy, right?
So the crucial point is knowing exactly who these microscopic MVPs actually are. After crunching all the data from the petri dishes, the seed inoculations, and the seedling trials, 3 absolute standout champions emerged. And it turns out they're all fungi. We're talking about Penicillium citrinum, Silaria curta, and Clonostachys rosea. These 3 consistently blew the competition away across all the tests, suppressing the disease while also boosting plant growth. And, fun fact, this study is actually the very first time Penicillium citrinum and a bunch of its close relatives have ever been recorded successfully inhibiting a Phytophthora species. Huge win.
The implications of all this are just incredibly exciting. By simply taking a healthy plant's natural internal bodyguards and putting them back into the farming ecosystem, we are looking at a future with vastly healthier cinnamon plantations. I mean, for the farmers, this means preserving their entire livelihoods against a really devastating rot. For the environment, it's a massive win because it slashes the need for those expensive, harsh chemical sprays. And for you and me, it means 0 toxic chemical residue on that cinnamon we sprinkle on our morning lattes and bake into our favorite treats.
It's just a perfect reminder that sometimes the absolute most powerful solutions to our biggest, most daunting agricultural problems are completely invisible to the naked eye, which leaves us with a truly exciting thought, really. If these naturally occurring microscopic bodyguards can save the world's cinnamon supply from total collapse, what other vital global crops could we protect just by finding the right microscopic partner? Nature has literally been perfecting these defenses for millions of years. We just have to be curious enough to look inside. Thanks so much for joining me for this explainer, and as always, keep questioning the world around you.